My name is Bryan Reatini. I am in the biology department at UNC and I'm in my 5th year of my PhD. The primary challenge that my research aims to solve is the identification of threats to local biodiversity in the Galapagos Islands. In particular, I'm interested in investigating the threat of interactions between native and invasive species. And the interaction that I've been specializing in is when those two species are occurring in the same area if they actually mate with one another. And the reason why that can be problematic is that you can imagine if you have a native flower and an introduced flower in the same location and they're pollinated by a bee, let's say, since they're distinct species, the offspring that they produce might be problematic. Those seeds might be dud seeds, or they might not be able to germinate or grow well. And this is a problem if you are that native flower, because some of the reproductive effort that you've invested in creating that seed has been wasted. The species pair that I've been focusing on for this work has been a native and invasive species of guava tree. So I've been investigating this negative interaction between these two, and as it turns out, it looks like the native populations of guayabillo do appear to be collapsing in areas where they're surrounded by these invasive individuals. And so this phenomenon of failed hybridization appears to be contributing to the local extinction of this native species. The Galapagos provides an archipelago with multiple different islands where different ecological and evolutionary processes may be occurring independently on those islands. And so, for example, with this species of guava that I'm studying, this invasive species has been introduced to the 4 human-inhabited islands at 4 distinct time points. And as a result, the process of that invasion facilitating the decline of the native species has been captured at 4 distinct time points along that process. And so instead of typically being limited to our single time slice when we go in and evaluate how processes are playing out, we actually have 4 distinct time points. And in having those time points, we can more thoroughly evaluate how does a process like local extinction by hybridization occur from beginning to end. The global impact of my research is that this specific phenomenon that I've been evaluating between these specific species in Galapagos might actually be occurring widely across archipelagos around the world, such as Hawaii, New Zealand, the Canaries, etc. And the reason for that is that archipelagos have been subject to an alarming number of species introductions. In fact, they show the greatest rate of species introductions relative to other ecosystems worldwide. And as a result, species introductions represent one of the principal threats to native biodiversity. The Galapagos Science Center has enabled this research in a number of ways. First, it has really smoothened out the permitting process, which there's no way I would have been able to do by myself. It has also provided financial support in that I received a seed grant from UNC and the GSC that enabled some of the research that I would have otherwise not had the funding to do. I was fortunate enough to have a Fulbright grant and be able to stay down in Galapagos for a full year. However, the seed grant is what enabled the actual specific research activities, such as visiting some of these isolated islands and being able to get to some of the populations that I otherwise would have never been able to work with. In addition to financial support, the GSC, the people at the GSC have really made this research possible. The folks specifically in the microbiology lab, my research involves genetic and genomic research, and so I spent a lot of time in the microbiology lab in order to do DNA extractions and work hands-on with the samples that I was collecting. That lab is full of great people and great supplies, and without that I would have never been able to accomplish this.